In this video, we're going to talk about secretion, clearance, and micturition. Hey, what's up you guys, and welcome back to my channel. So today's video is going to be the last one before we get to talk about fluid and electrolyte balance. I know, it's pretty exciting. So today, we're going to take a look at the last step in the urine formation process. We're going to talk about how we can estimate kidney function, and then we'll look at the excretion of urine from our body. So if you have a full bladder, this is your cue to go empty it, then come back, grab a notebook, and we'll get started. So we saw last video that reabsorption depended on membrane proteins in the membrane of the nephron, while secretion also depends upon transporters. But in the case of secretion, we are talking about substances moving from the plasma into the nephron. In this way, secretion is similar to filtration. However, unlike filtration, secretion occurs later in the nephron and not at the Bowman's capsule. Plus, our body decides what we secrete and not necessarily what we filter. And in this way, secretion is a more selective process than filtration. And at the same time, we can actually excrete extra amounts of a substance through secretion. So say something gets filtered. If that thing is not reabsorbed, then it's bound for excretion. And our body can actually choose to excrete extra amounts of that thing by secreting that thing from the plasma into the nephron. So now, what is filtered gets excreted, and what is secreted gets excreted. And in plain terms, we can put more of a substance into the filtrate to get excreted as urine. And this process we call secretion. And we will talk about electrolyte and ion balance in later lectures, but the kidneys secrete hydrogen ions and potassium ions in order to maintain homeostasis. So recall from one of our previous lectures that we had a formula that talked about what gets excreted. So excretion equals filtration minus reabsorption plus secretion. But this formula doesn't tell us how well our kidneys are working, because it doesn't tell us solely how well we are filtering things, because filtration is the true measurement of kidney function. And in order to assess kidney function through blood and urine samples, scientists use the concept of clearance. So clearance is the rate at which a substance vanishes from the blood. Another way to look at clearance is that it is the volume of plasma needed to pass through the kidneys in a certain time to clear that substance from the body. And clearance is measured in milliliters per minute. Didn't we see something else that was measured in milliliters per minute? GFR, right? That was measured in milliliters per minute. So here's the interesting thing. Clearance equals GFR under certain conditions. The first condition is that the substance we are measuring must be freely filtered. And by freely filtered, I mean 100% of what is in the blood gets into the nephron. The second condition is that there must not be any reabsorption of that substance. And the third condition is that there must not be any secretion of that substance. And all three of these criteria have to be met in order for clearance to equal GFR. So inulin is a polysaccharide that scientists use to estimate GFR in lab settings. And the reason that they use this substance is because it's freely filtered and neither secreted nor reabsorbed. And so its clearance equals GFR. So you might be thinking to yourself, why do these three criteria have to be met? So in order for clearance to equal GFR, we said that it has to be freely filtered, and it cannot be secreted, and it cannot be reabsorbed. So that means everything that we filter equals the amount that ends up in our urine. And if everything that we filter ends up in the urine, then we can just look at our urine to see what our clearance and therefore our GFR actually is. And the reason that we look at our urine is because we can't cut open a nephron to see what our filtration rate actually is. Cool? Okay, if you have any questions, then drop them below because I'm moving on. All right, so let's look at a formula related to clearance. So clearance equals the urine concentration of a substance measured in milligrams per milliliter times the urine flow rate measured in milliliters per minute divided by the plasma concentration of that substance measured in milligrams per milliliter. Okay, now I know we talked about inulin and how it's used to estimate clearance and therefore GFR, but in real life, we don't use inulin to measure clearance. 1, because it doesn't naturally occur in the human body, and 2, because it has to be administered by continuous IV infusion. So we use something a little different in real life settings. We use creatinine. So creatinine is a breakdown product produced by the muscles, which means it's naturally produced by the human body and we don't have to administer it. And so we can measure a serum creatinine level in your blood, and that's just a measurement of how much creatinine is in your bloodstream, and it correlates with GFR. So if serum creatinine is really high, this means that GFR is really low. And kidney injury and kidney disease are really the only 2 things that can elevate your serum creatinine levels. And serum creatinine only elevates once 50% of kidney function is lost. Now, all substances have a clearance rate. It's just that inulin and creatinine are the only substances whose clearance rate equals glomerular filtration rate. And just to make sure that we understand this concept, let's look at some examples. So when we talk about clearance, let's use an example of amino acids. Amino acids are a very hot commodity, and our body wants to hold on to these as much as possible. So the plasma concentration of amino acids is high, but the urine concentration of amino acids is low, because like we said, we do not want to excrete these. So mathematically, if you look at this formula, then clearance of amino acids is low. The plasma concentration of amino acids is inversely related to the clearance, and the urine concentration of amino acids is directly related to the clearance. But on the other hand, ammonia is a waste product, and we don't really want it in our blood. So the urine concentration of ammonia is high, but the plasma concentration of ammonia is low. So mathematically, the clearance of ammonia is high. We are probably secreting extra amounts of ammonia into our nephron in order to excrete it. Clearance may be so high in this case that it's higher than glomerular filtration rate. And actually, when we secrete a substance, our clearance rate of that substance will be higher than GFR. Can you tell me what it would look like if we reabsorbed a substance? So if we reabsorb amino acids, then the clearance of those amino acids would be lower than GFR. We are filtering more amino acids than we are excreting, because we need to reabsorb those amino acids in order to keep them in our body. Okay, so for the last part of our lecture today, we're going to talk about micturition. And micturition is also called urination. So once fluid leaves the collecting duct, it goes to the renal pelvis and we call it urine. And from the renal pelvis, it goes through the ureters to the bladder. So the bladder is a hollow organ that's composed of smooth muscle, and the urethra is held closed by 2 sphincters. The internal sphincter is composed of smooth muscle, and the external sphincter is composed of skeletal muscle. Okay, so the external sphincter is held closed by tonic stimulation, and it is always contracted unless that tonic stimulation is inhibited. And we inhibit that tonic stimulation when we urinate. So micturition is a spinal reflex. First, our bladder must fill up. Secondly, stretch receptors on the bladder wall must send a signal to the central nervous system. The central nervous system then tells the parasympathetic neurons to fire. Parasympathetic neurons contract the smooth muscle in the bladder wall and increase pressure in the bladder. This increase in pressure opens the internal sphincter. Somatic motor neurons going to the external sphincter will be inhibited. Then urine will be able to pass out of the bladder to be excreted. So in the case of a person who is toilet trained, there are additional sensory fibers in the bladder that send a signal to the brain and tell the brain the degree of bladder fullness. The central nervous system will inhibit the parasympathetic neuron and double down on the contraction of the external sphincter until an appropriate time to urinate arrives. And for us nurses, that could be 13 hours, right? Hey guys, welcome back. So I hope you understood that part about clearance. I actually thought that it was pretty self-explanatory until physiology books throw in all of these formulas. So don't hesitate to rewatch this video or other helpful videos that you might find. If you got value out of this video, then go ahead and hit that like button. Comment down below if you have any questions or want to see specific content. And don't forget to subscribe for new videos posting every week. Otherwise, stay safe and I'll see you guys next time.